Okay, so now that we've introduced the major players to our nutrition chapter, let's talk a little bit about what we're actually doing with them. Something that we've talked about a lot, I don't honestly know if we've ever really defined it or not, but is the idea of metabolism, right? And a metabolism is just all of the biochemistry happening inside of your cells. And really biochemistry takes two forms for us. And we've talked about these forms way back in A&P 1, but it's worth kind of revisiting them now. Other things that are building, right? So we make large things out of small things. This is anabolism, right, or anabolic processes, right? So when we talk about making a protein out of amino acids, right, it's an anabolic process. And the other sort of form, obviously, is the opposite of that, where we take a big thing and we break it down. So if you remember when we talked about hydrolysis and synthesis before, we talked about adding a water molecule to something to break it apart, to take a water molecule away from something to stick things together. This is what that comes down to, right? Anabolic building, catabolic breaking apart. And of course, remember also here what this means for us in terms of energy. When we make things, we stick them together, right? We're forming bonds, we're storing energy. We break them apart, we're breaking bonds, we're releasing energy. So again, this can be a process in terms of managing an energy budget here as well. One of the things that we're going to do a lot in terms of an energy budget is shifting around phosphate groups. Okay, ATP, right, adenosine triphosphate, is a amino acid with 3 phosphates stuck to it. And the reason we use that as fuel or as a fuel storage source is that those bonds, those phosphate bonds, have a lot of energy associated with them. Okay, and so an ATP has 3 of them, right, that we can use to do things around the cell. And we do so by giving that phosphate group to something else, right? Process that we call phosphorylation, right? And when we phosphorylate something, when we give it one of those one of those phosphate groups, we give it energy to do what it needs to do. Okay, so how does all this then apply to what we're talking about now, what we've talked about very recently? The processing of nutrients here is basically this 3-step thing for us, right? So we begin with what we talked about in the digestion chapter, right? We bring food in, we break it down into its nutrients, we absorb the nutrients, and we send them around in the blood to where they need to go. Okay. Once they're where they need to go, once they're in the cells they need to be in, right, we can use these things to build or to break down, depending on our needs. Right. One of the major processes in all of this sort of metabolic nightmare is what we call cellular respiration, right? The oxygen-related production of ATP from glucose. And again, we've talked about some of these steps along the way, maybe not in as much detail as we're going to now, but hopefully some of this will at least be a little bit of review. And the processes here in cellular respiration include some of, no, not necessarily all of, glycolysis, the Krebs cycle, and this oxidative phosphorylation step. The one that we can skip is glycolysis. And the reason for this is that if we're going to use something other than glucose as our initial fuel, if we're using, say, a protein, right? If we're using a fatty acid, okay, we're going to skip this part and we're going to stick the component that we're using directly into this Krebs cycle. So what does all this look like? All right. So here the most common way, and the one we're going to start with, is carbohydrate metabolism, which is starting here and working our way down all the way through here. Right. Basically, the idea here is that we take our carb and we break it down into glucose. So if we start with glucose, right? We can go right in here. If we have a different form, fructose or whatever, we have to convert it to glucose first, whatever. And then we can go through the process of glycolysis, where we break that down into pyruvic acid. We convert that pyruvic acid into this guy right here, acetyl coenzyme A. And I'm going to highlight this guy right now because acetyl coenzyme A is super, super important. And the reason it's so very important is that it is the converging point for all three of our metabolic pathways. Proteins, carbs, and fats all come down to acetyl coenzyme A at some point. Okay, we take this acetyl coenzyme A, we toss that into the Krebs cycle, we convert it into a form that we can then move on to the last part here, oxidative phosphorylation. So let's start here with this middle pathway, carbohydrate metabolism. We turn glucose, which is this happy little guy right here, C6H12O6, and we take this guy in the presence of oxygen, we turn it into water, so 6 waters, 6 carbon dioxides, 32 ATP, and some heat, of course. You'll be aware, of course, that this and this balance out, right? They have to balance out, otherwise it doesn't work. This pathway, right, in terms of its complete pathway, involves all three of these stages: glycolysis, Krebs, oxidative phosphorylation. What does this end up looking like, right? Well, along the way, we take our glucose and we break it into pyruvic acid, 2 pyruvic acids. This is glycolysis, all right? That pyruvic acid gets converted to acetyl coenzyme A. We dump it into the mitochondria where the Krebs cycle occurs, okay? We convert this into a form that we can pass off into this secondary pathway. In terms of the ATP that comes out of this, right, we net 2 from glycolysis, we net 2 from the Krebs cycle. We get somewhere around 28 from electron transport. Now, I'm not going to make you guys worry about the products of all of these things, NADH and FADH, NADH2 and FADH2, so on and so forth. I'm not going to make you guys worry about that stuff. That's for a different course altogether. Right now, I'm mostly concerned with you remembering the steps, right, and the ATP produced from each one, which is really what this is all about, right? Complete breakdown of 1 glucose through this pathway nets carbon dioxide and water as our waste products, 32 ATP, right, in total. Now, again, there's some uncertainty here about exactly how many comes out of this, 28 to 32 to 34. Sometimes you'll see the number 34 tossed around. We're not exactly sure. Obviously, this is not the sort of thing that we can like measure in person. And so the chemistry is still a little bit questionable, but it's a lot of ATP at the end of the day. 32, 30, these are good numbers to kind of keep in mind. Now, the opposite version of that process, right, where we take glucose and we break it down to make energy, are various storage mechanisms, okay? And the first storage mechanism that we encounter is when we have too much glucose and we want to kind of put it away maybe for an hour or 3 hours or a couple of days later, right? We're going to do that by turning it into glycogen, which is just a bunch of glucose slapped together, right, to form one larger molecule. The process by which we make glycogen from glucose is glycogenesis, all right? When we do this, we take excess glucose, we turn it into glycogen, we store it in the liver, we store it in the muscles too, but mostly in the liver. And then when we need it later on, right, say, oh, now we're out of glucose, let's take some of that stuff out of the pantry, break it down in glycogenolysis, right? We turn glycogen back into glucose, which is kind of these two processes here, right? We kind of shift between glucose and glycogen through gluconeogenesis, not glycogenolysis here, breaking it down, and glycogenesis to make it. One of the reasons that becomes important for us, sort of maybe clinically, is with carboloading, right? So glycogen, because it's a fairly simple mechanism for the storage and release of glucose, is very, very useful for us if we're going to be doing something high intensity in terms of activity, right? We know that in the future we're going to need a lot of ATP quick. Our easiest source for that is glycogen, and turning that back into glucose. So the process of carboloading is basically just eating way too many carbs for 3 or 4 days before you're going to have your athletic event. Okay, sitting on your butt a lot. Okay, and here what that's going to then do for you is you're going to have, on the day of the event, massive glycogen stores, massive readily available glucose that you can perform at a higher level for longer. It doesn't work outside of that 3 or 4 day period though, right? For long-term storage, what we're going to do is actually turn that glycogen into fat, and you can't really access that fat particularly quickly. So there's a limited window under which carboloading is going to be valuable for you. Last big major process here in glucose metabolism, carbohydrate metabolism, is gluconeogenesis. And this is when we take pyruvic acid and slap it back together, right? So remember, we take glucose, we break it down into 2 pyruvic acids, right? We can also take 2 pyruvic acids and turn it back into glucose. We can also take glucose and manufacture it from waste products of, well, not waste products, but the breakdown of fat. So glycerol in particular can be turned into glucose. Basically, the idea here is we're manufacturing new glucose, right? New neogenesis, making glucose when our blood glucose levels are dangerously low, right? And again, one of the problems with that is that your nervous system can't do anything other than use glucose. So you can die, which is an option, right? Or you can kind of cobble together some emergency glucose from spare parts when you need it. That's what this is all about. Okay. Our second most common metabolic pathway is lipid metabolism. So we're using lipids here to manufacture energy in some way, shape, or form. Now, as it turns out, there's a greater energy yield from fats than from anything else, right? 9 kilocalories per gram versus 4 from carbs. You think, well, why the hell would I not use this all the time? The reason that you don't use this all the time is that it's a pain in the ass. It is not simple in the way that glucose metabolism is. Okay, so it's a lot more steps. There's more mouths to feed along the way. There's more things to do. It's just simply not the increase in energy efficiency is not made up for by the complication of the process, I guess is what I'm saying. If it were a simpler pathway, we would absolutely use fats before we ever use glucose. In terms of what we're going to be doing here, again, we have to go back and remember how a lipid is put together, how a neutral fat is put together. And for that reason, we can simply kind of omit cholesterol from the pathway here. We're only going to use these triglycerides. Because what is a triglyceride? It's a glycerol molecule with 3 fatty acids stuck on it. And we can convert all 4 of those things into energy in some way, shape, or form, although we're going to do it separately. So the first thing we're going to do is we're going to snap these 3 fatty acids off of our glycerol. And we're going to take that glycerol and we're going to turn it into pyruvic acid. Okay, so if you remember, from 1 glucose, we got 2 pyruvic acids, right? And we tossed that into the Krebs cycle and we made a bunch of ATP out of it, yada, yada, yada. We only get 1 pyruvic acid from 1 glycerol. So a glycerol gives us half the amount of ATP the glucose molecule does. So instead of getting 30 or 32 ATP, we get 15 or 16. Right? So it's still a lot, right? That's still not nothing, but it's not as efficient. The fatty acid pathway, on the other hand, this is where, again, life is a little bit more complicated. We take the fatty acids, we break them down into little chunks, okay? Essentially, what we end up doing is you make a bunch of little acetic acids out of the whole fatty acid, one fatty acid. You convert each of those acetic acids into acetyl coenzyme A, right? That middleman. Then we can toss that in the Krebs cycle. So, in diagrammatic form, right? Here's our neutral fat. We break them apart, process called lipolysis, right, into the glycerol and the fatty acids. The glycerol gets converted to pyruvic acid, 1-pyruvic acid, and dumped into this process. The fatty acids get broken down into acetic acid and then dumped into this process here. So there's just more work, really, than we need to really do. The other part of that whole process that makes this maybe not as desirable is that the conversion of fatty acids to acetyl coenzyme A results in the production of ketone bodies, which can result in ketoacidosis, which can kill you. So obviously anything that has a potential for killing you is not a preferred option for us, just in general. The other idea here is not the breakdown of carbohydrates, or not breakdown of carbohydrates, the breakdown of fats into ATP, but the conversion of excess carbohydrates into fats. And this is lipogenesis. Okay, so we can, if you look back at our diagram, right, yes, we've got all these blue arrows here talking about catabolic or sort of breakdown pathways here would make energy out of this. There's also these purple arrows that go the opposite direction. Right? There are also anabolic pathways that lead back to the production of triglycerides. And that's what lipogenesis is. Basically, because we have this middleman pathway, right, this acetyl coenzyme A pathway, in all of our metabolic pathways, we can use that to kind of go back and forth between different major nutrients, right? We can very, very easily then convert excess carbohydrates into fats. That is what lipogenesis is. This is what all of this is talking about in this slide. If you remember that basic idea, I'm happy with that. The opposite part of this, what we've talked about before, the breaking down of the triglyceride into the glycerol and the fatty acids, is lipolysis. That's all that is. Whether you remember this part here, oxaloacetic acid, not so worried about it. Finally here, to wrap this particular part up, again, I just want to really emphasize here, ketosis, ketoacidosis, not great, not a good idea, okay? As you burn fats through exercise and proper dieting, you will accumulate ketones in your blood. It happens, right? The ultimate goal here, however, is not to have them build up. If they build up slowly over time to the point where you can properly get rid of them in your urine, everything's good, okay? But if you put yourself on a starvation diet or any of those keto diets you see advertised in those shitty grocery store magazines, you can put yourself in a position to die. And I don't want to spend too much time talking about that. I will put a link in the description of this video to a little more explanation of why ketoacidosis and keto diets are bad and why they'll kill you. But we'll leave it there for now. In the next and final part of our lecture here, we'll kind of continue on and we'll finish off, particularly with things like cholesterol balances and stuff like that, the last kind of little vestiges of what's left here for nutrition.